Thank you, both of you, very much. We are not going to have time for Q&A right now for these two panelists, but if we pick up time later in the morning, we will come back to any specific questions for these two. We're going to move on now. Thank you very much, Dr. Espinal, Dr. Rosenberg. We're now going to move on to the session on virus vectors and reservoirs. Scott Weaver will be holding down the fort as the one person who is here in present and who is present in the room, and one or both of the other speakers will be joining remotely. Scott Weaver is the John Seeley Distinguished University Chair in Human Infections and Immunity, University of Texas Medical Branch. Tom Monath is Chief Scientific and Chief Operating Officer, Bioprotection Systems Newlink Genetics Corporation. And Dwayne Gubler, who is in Vietnam, is Professor and Founding Director, Signature Research Program in Emerging Infectious Diseases, the Duke National University of Singapore Graduate Medical School. And please join me in welcoming Scott Weaver.
Well, thank you, Mary and Diane and the Academy for the opportunity to participate in the conference today, and everyone for braving the weather to come out. And I'm going to begin by pointing out what an African virology friend of me told me a few weeks ago. Not only do we not know very much about Zika virus, but for the past half century, we've gotten the name wrong. The original spelling from Uganda has 2 I's, and we've been using 1 all this time. I think it'd probably be a bad idea to change to 2 I's at this point, so we'll stick with 1 for now. Dr. Rosenberg went through some of this information, the discovery of Zika virus in the 1940s when the Rockefeller Foundation began yellow fever enzootic studies and also describing many viruses in that part of the world. The virus was isolated for the first time from a caged sentinel rhesus monkey hoisted up into the canopy, and it became viremic and developed a fever, and virus was isolated in suckling mouse brains. The following year, the virus was isolated from Aedes africanus mosquitoes, also collected up in the canopy, and then later 3 human infections first detected in Nigeria. And then in 1956, there was actually an experimental infection done with a human volunteer that confirmed the relatively mild flu-like illness typically caused by Zika virus. And then the first isolation in Asia came in 1966 from Aedes aegypti collected in Malaysia. And then finally, the first human infections in Asia in 1977 in Indonesia. So what we know about the distribution of Zika virus over that long period of time is shown in this map. In red are the countries where there's evidence from serosurveys that people have antibodies against Zika virus. But I need to point out that many of these studies, especially the early ones, were done with very non-specific methods that may have cross-reacted with, at that time, unknown flaviviruses. In gold are the locations where the virus was detected, either in human cases, mosquitoes, or reservoir hosts such as monkeys. So it was clear for many years that the virus was present both in Africa and in Asia. Now I'm going to talk about the enzootic cycle of Zika virus in the context of studies done since the 1970s in eastern Senegal, where what I like to call a quartet of arboviruses circulate in a virtually identical sylvatic or enzootic cycle in gallery forests in that part of West Africa. And the studies in Senegal have shown that there are 3 mosquitoes that are important vectors in this cycle: Aedes furcifer, Taylori, and Luteocephalus, that occupy forests. And then there are 3 principal non-human primates that have been identified as amplification hosts. Those include Potus, African green monkeys, and Guinea baboons. And these viruses generally appear in Ketagoo only every 7 to 8 years when large numbers of mosquitoes are sampled. But the exception in the quartet is Zika virus, which appears much more frequently. And this suggests that there may be other shorter lived reservoir hosts with faster turnover rates in this part of Africa for Zika than for the other 3 viruses. Another factor could be transavarial transmission, which has been documented by the isolation of Zika virus from male mosquitoes in these forests. We think that the main mosquito that can transport this virus to nearby villages is Aedes furcifer, because our field studies have shown that it leaves the forest and enters villages to bite people. But actually how the virus then would move from a nearby village or people entering the forest to an urban habitat where it can establish the urban cycle is not well understood at all. But during the urban cycle, various mosquitoes like Aedes aegypti and Aedes albopictus in the subgenus Stigomyia apparently serve as vectors. So I'd also like to look a little bit at the history of this quartet of viruses, their history of emergence from this enzootic cycle into human transmission. So yellow fever virus, I think we understand fairly well. Like Zika, we think it originated in Africa. And then during the age of sailing ships, especially during the slave trade, these ships would transport both domesticated Aedes aegypti, as well as people who are susceptible to infection, and an onboard transmission cycle would carry the virus across oceans into the New World, and we had regular outbreaks in various tropical locations, even in temperate locations where the mosquitoes were introduced and could survive for the summer in the northeastern US. But as far as we know, this never happened in Asia, and it's not clear why. Dengue viruses, we think, originated in Southeast Asia, where the 4 or 5 serotypes diverged when they were still in the enzootic primate cycle. Then they independently moved into human transmission cycles and probably were also transported around the world by sailing ships. We also know that dengue virus subtype 2 apparently spilled back from that transport into forests in West Africa, where, as I showed you before, it circulates today in an enzootic cycle. But as far as we know, this has never happened in the Americas. There's no clear evidence of enzootic transmission. And finally, chikungunya virus also has a long history of emergence from Africa, probably aboard sailing ships during the 18th century to both Asia and to the Americas. Same kind of scenario as yellow fever and dengue. But there's no evidence that enzootic transmission was ever established in Asia or so far in the New World either, although it's only been here for about 2 years now, at least in recent history. So what's the risk for spillback of Zika into an enzootic cycle, either here in the Americas or elsewhere in the world? Well, again, if we look at this quartet of viruses and look at this in a very simplistic way, how often has this happened with these other viruses? So yellow fever, it has succeeded to establish enzootic cycles in the Americas, but not in Asia. We're not even sure it's ever been transported to Asia, although the history of chikungunya, with multiple introductions from East Africa into Asia, suggests the opportunity has always been there. Dengue viruses, we know that dengue 2 has spilled back into an enzootic cycle in West Africa, but so far not in the Americas, so 1 out of 2. Chikungunya, as far as we know, has not established an enzootic cycle anywhere outside of Africa. And then for Zika virus, there's no direct evidence of enzootic transmission outside of Africa, but of course there's been virtually no surveillance done for Zika or even for many of the other viruses in most parts of Asia or even parts of the Americas, so the information is very limited. And the problem is that defining or proving the existence of these enzootic cycles is logistically extremely challenging. Unless you can find viruses that are genetically very distinct, coming from forest-dwelling mosquitoes versus urban mosquitoes and people, like we have done for dengue, it's very hard to tell whether there's an enzootic cycle unless you detect it by an epizootic in monkeys, like we see in the New World with yellow fever virus, for example, or you work in very remote locations. So this is a phylogenetic tree, the most recent genomic strains of Zika virus available through GenBank and our own work in Galveston. And you can see that there are two main lineages of Zika virus, one found in Asia and recently in the Americas, and then a second one appears to be restricted to Africa here. And here are some of the recent strains from Asia and the South Pacific, and then the American strains up here. I want to point out here the main finding that, first of all, the closest relatives in the flavivirus genus are African sister viruses of Zika, suggesting that Zika probably did originate in Africa. The remainder of the conclusions from this work I'm going to show you in a map format here, showing the history of spread as far as we can deduce from these phylogenetic studies. So we think that the virus may have been introduced into Asia a long time ago, as early as centuries ago. It's impossible to distinguish that scenario from us just not having sampled another enzootic lineage in Africa that gave rise to the Asian strains and would be more closely related. So we don't know for sure. Then in 2007, as Dr. Rosenberg showed, the virus was introduced into yap, caused an outbreak. The same year, there was an outbreak in Gabon. This one was clearly transmitted by Aedes albopictus mosquitoes, the yap by Aedes henselae. Then the virus spread into French Polynesia and on to other areas of the South Pacific. And we think then that the next introduction into the Americas probably occurred from French Polynesia. But I also want to point out that the yap and the French Polynesian introductions appear to be independent, and they probably came from some part of Southeast Asia where we simply aren't doing the surveillance to find the progenitor strains. And then, of course, subsequent spread, as he just said, throughout Latin America and the Caribbean now. And then finally, a recent outbreak in the Cape Verde Islands off the coast of Senegal. And I was told informally by an African virologist that they think that this strain originated directly from West Africa. And it appears to be transmitted by Aedes aegypti. So why did the sudden Zika emergence occur during the past few years? I'm just going to present a few hypotheses that, in my view, are some of the leading ones. There are certainly others. This is not an exclusive list. One is that Zika virus underwent some sort of adaptive evolution to enhance its infectivity of these Aedes stegomyia mosquitoes that are transmitting in all of these recent outbreaks. And the precedent for this hypothesis comes from chikungunya virus, which in a series of very detailed studies we now understand has, between 2005 and 2009, it underwent a series of adaptive mutations in its envelope glycoproteins that allowed it to infect Aedes albopictus much more efficiently, and this allowed it to expand its geographic range into places like Italy and France and many parts of Africa, where Aedes aegypti does not occur. The second hypothesis is that the adaptive evolution did not involve mosquito vectors, but it involved the human host, and perhaps increased efficiency of infection of humans and replication could lead to higher levels of human viremia, which would also facilitate more efficient circulation of the virus and spread, and also could explain the higher risk of transplacental fetal infection. And finally, the remaining hypothesis is that nothing at all has changed with this virus. In fact, possibly all Zika virus strains from Africa or Asia have the same epidemic potential, but by chance the virus was introduced into naive areas with the right numbers and populations of susceptible people and vectors to start the amplification, and especially in French Polynesia, where the populations were large enough to reach the levels of tens of thousands of infected people, that may really have, for the first time, increased the risk of the virus dispersing around the world in infected travelers and establishing itself in the most naive population on the planet, which is in the Americas. And the Cape Verde epidemic, where it looks like we're seeing the same kind of epidemic transmission through Aedes aegypti, supports this hypothesis that maybe the African strains as well have this epidemic potential. So why didn't we detect outbreaks of Zika before? I showed you evidence that it's been in Asia and Africa for many centuries probably, yet we've only detected 14 infections prior to 2007. Well, it's possible that epidemics have occurred in those locations, but they remained undetected because of the difficulty in making a clinical diagnosis, distinguishing from dengue, chikungunya, or even yellow fever, and the lack of specific diagnostics for Zika virus in particular. And probably very few labs in the world have ever even tried to test human samples for Zika prior to the last few years. Another hypothesis is that there's a relatively constant level of either spillover from enzootic cycles in Africa, perhaps also in Asia, that result in relatively constant levels of infection in people throughout their life, so that there's a regular occurrence of Zika cases, but we simply don't even notice them because this is in a background of malaria and chikungunya and dengue and so many other infectious diseases in places like Africa and Southeast Asia that we don't notice them unless we specifically go looking for them. And under these circumstances, the herd immunity, and this is actually supported by some of the seroprevalence I mentioned earlier, which can be as high as 75% in Asia and Africa, that herd immunity may even protect women by the time they reach childbearing age, at least some of them, from the risk of microcephaly. So if there's a small number of constant infections going on and a lower rate of microcephaly, microcephaly may occur at a frequency only slightly above the levels we see for other viral infections and never hit anyone's radar screen at all until we see a major epidemic. And so Zika virus strains in Africa and Asia perhaps didn't need to undergo any kind of adaptive evolution and may not have, or they may have lower efficiency of transmission depending on which of these hypotheses you want to believe and test. Finally, I just want to acknowledge the people who have done the Zika work in my lab group shown here. Our arbovirus group in Galveston, the evolutionary studies, Rubing Cheng, Bob Tesh, and Nikos Vasilakis especially. And then this is the group that did the field studies in Senegal, including from the Institut Pasteur, Amadou Sall and Malouf Diallo, Kathy Hanley, Derek Cummings, Ben Elhaus, and Doug Watts also. And we had very important support from the NIH, especially from our program officer, Christina Cassetti, who really allowed this project to take place in eastern Senegal. Thank you for your attention.